Go ahead.
According to scientists Dudna J. and Charpenter in the magazine Science, the ability to engineer DNA in cells and organisms easily and precisely will have major implications for basic biology research, medicine, and biotechnology. We know that scientific breakthroughs happen every day, but did you know that researchers now have the potential to alter DNA and change our bodies and functions? Hello, my name is Isaiah Rodriguez, and today I want to talk to you guys a little bit about what genetic research is and why it's important. I've written multiple research papers on this subject, and I've researched a lot of articles on the ethics of human genetic structure and what it means for us. The genetic alteration is important to our society today. Using CRISPR and other gene-changing methods, there are many benefits and downsides to gene changing, many of which could affect our daily lives significantly. Before we begin, I'd like to talk a little bit about what CRISPR is and why it's important. You see, CRISPR is a gene-changing method. Scientists use this method to easily see how genes are altered and how they affect living things' DNA. CRISPR isn't a tool, but it's actually a series of steps that researchers use. Now what they do, according to Cambridge University, is they insert a little protein into the DNA of a patient. And then the protein cuts a piece of the DNA off, and once they cut the piece of the DNA off, they can insert a little part of what it is that they want changed. Now, it's a bit hard to explain since we really can't see it in action, but it's basically like cutting off a piece of blanket and then patching it on with new material. The protein is the scissors that you guys use to cut the blanket off, and the DNA that scientists want to go on there is the new piece of material. It sounds easy, and that's because it is easy. It's so easy now for researchers to alter DNA, in fact, that it has made them take a step back and say, whoa, we have to slow down before something bad happens and we don't even realize it. Researchers who published in the Nature Journal of Science, Lanfer et al., said that there are, quote, grave concerns regarding the ethical and safety implications of these experiments. Basically, they firmly believe that the next plan is to alter human embryos, or babies, and that if it goes bad, there could be severe consequences to entire generations of people. Now that we discussed why CRISPR and gene editing are easily done, let's look at what the benefits are. There are multiple benefits for this type of research. This could cure many diseases like HIV or AIDS or maybe even some types of cancer. So when I say cancer, it could mean like the genetic makeup of a human embryo, and the scientists could alter the DNA of the embryo, and if they spot the pattern, that's susceptible to the cancer. Just like that, they've spotted a pattern, and then the person won't have to develop the cancer anymore because they've changed the way the pattern works. They must first identify the patterns that could lead to these diseases and then change them if they spot them, like using a computer program, for example. According to gene researchers Chen et al., there is also a lot of potential to profit from altering genetics in plants and crops in agriculture. They've done the experiments and predict that by making a certain type of gene, using CRISPR, more or less dominant, they could change the way the agriculture is kept and maintained in those businesses in a lot of ways. It will help the economy in our society as well. We've discussed the benefits, but now let's look at why this field of research can get really worrisome really fast. You see, CRISPR gene editing has given researchers the power to do some controversial things, like make human-animal hybrids or chimeras. In addition, the research can answer some issues that have been raised by other scientists who are in this field as well. Researchers who have already done a lot of experiments on human-animal hybrids, Fang et al., say that we already have the potential to grow entirely new human embryos with hybrids of animals, and then use the organs within these human-animal hybrids to give to organ donors or patients. The researchers don't know how to define the value of life in those babies and whether or not giving organs to humans is actually ethical or not. Scientists also aren't sure how altering stuff like DNA will affect our protection against other diseases, but they're doing it anyway. A lot of questions can be raised on this research, but there really aren't enough answers to provide support for the means that they are doing. A scientist named Hemmering wrote a journal on why we need to discuss more about this topic before research can go on. It might be easy to alter genes now, but once you alter them, it pretty much is irreversible since you do it, since you can only do it once at a certain point. In conclusion, genetic alteration is important to our society today, using CRISPR and other gene-changing methods. There are many benefits and downsides to gene changing, many of which could alter our daily lives significantly. CRISPR is important to the gene editing process and has made it easier. There are multiple benefits for this research, but there have also been some serious problems associated with it. To close, I leave you with a quote from a man named Aldous Huxley, who wrote a book called Brave New World, and said some things more than half a century ago that we can still think about today. I quote, "The more science discovers and the more comprehension it gives us of the mechanisms of existence, the more clearly does the mystery of the existence itself stand out." Thank you.
Good job, Isaiah.
Good job.